then share my screen. And let's go to the beginning of this. All right, so we're going to continue our discussion on memory, and I want to kind of go into this question of where memories get stored. And that neural correlate of memory storage in the brain, where in the circuits of neurons in the brain a memory is stored, has been referred to as the engram. And what I want to point out, remember, I divided memories for you, at least in humans, into two categories, which I called explicit or declarative memories. Those are the memories you have conscious access to. And then implicit or non-declarative memories, such as, motor learning and habituation, conditioning, any type of activity that you learn. And those we're going to discuss separately. And part of our discussion is where these memories get stored. So the brain areas that are important in storing declarative memory, we got a lot of clues as to where these areas are from the patients that we discussed last time, patients such as Henry Molaison and Kent Cochrane, who had extensive damage to their hippocampal area. So just to give you an idea, and I'm going to come back and discuss the anatomy of the brain in a bit more detail in a later lecture, but I just want you to think of the human brain and think of these three different planes that you can cut the brain or the body with. So the plane that goes kind of straight down but cuts through front and back is called a medial plane or a median plane. I'm sorry, it's called a sagittal plane. It's not called a medial plane. The sagittal plane, if it's exactly in the center, it's called the mid-sagittal plane. If it's to one side or the other, it's called the parasagittal plane. If you then cut in the direction like this, that is called the coronal plane. And if you cut horizontally, parallel to the ground, if you're standing, that's called the horizontal plane. So what you see here on the top left is a mid-sagittal section of the brain. And if you look under the cortical areas in the deeper parts of the brain, you see the structures that are associated with a lot of functions that are developed in the vertebrate animals that have evolved very early on. And what we know is that some of these structures, if you damage them, they have extensive effects on memory. So the one that I want to focus on right now is this structure known as the hippocampus. So you can see kind of the tip of it here. And here you see a kind of a horizontal section of the brain, but looking at it ventrally. So you're looking up at the brain from the bottom, and you kind of see the hippocampus structure, which is bilateral sitting there. And I told you, as a three-dimensional structure, the hippocampus goes on the two sides, comes up, and it kind of turns around like a horn, like a ram's horn. And that's why it's called hippocampus, which means seahorse. Now, if you take the hippocampus and now take coronal section, so you come down here and take a section of the brain and look at a slice of the hippocampus, you see this kind of structure that is well preserved. This part of the brain, this is kind of, it's not the neocortex, but it's a very primitive cortical area in that it has layers. It only has 4 layers. Sometimes it's referred to as the allocortex or the old cortex. And vertebrate animals that are not mammals also have these types of cortex, which is allocortex, but they don't have neocortex. For example, fish and lizards and things like that, animals like that, they don't have a neocortex. Now, the hippocampus and the areas of the cortex in mammals, especially in humans, that are nearby the hippocampus, which are called the parahippocampal structure, they all sit in an area of the brain which is called the medial temporal lobe of the brain. Okay, so all of these structures that are part of the temporal lobe, the medial temporal lobe, are involved in memory formation, especially the hippocampus. And I'll come back and tell you a little bit more about the hippocampus in the evidence that we have. And part of the evidence in humans is actually very interesting, is from brain imaging studies. And this was a paper that came out at the turn of the 21st century in the Proceedings of the National Academy of Sciences, in which they studied using brain imaging the hippocampus in London taxi drivers. These are the years almost before we had GPS in all the cars. So if you wanted to become a cab driver in London, you had to have extensive knowledge of the streets of the London metropolitan area. And you had to pass this knowledge of London test. And that required you to know 320 different routes in the city, 25,000 individual streets, and about 20,000 landmarks in London. And people who studied for this test took 3 to 4 hours a day for almost a year to study to just become a cabbie. And then as they became taxi drivers and started learning more and more and more, they became better and better at the knowledge of these streets and landmark areas. Now, what these guys did in this paper is they looked in the brain of these taxi drivers, and they looked at sections of the hippocampus, and they measured the area of these cross sections of the hippocampus on the two sides of the brain and compared the taxi drivers versus controls. And what they found is that in the posterior part of the hippocampus, the volume of the hippocampus that they measured the volume based on these cross-sectional areas. The volume depended on the time that the person spent as a taxi driver. So this part of the hippocampus was growing bigger and bigger and bigger in people who were learning these different routes. Which again is indication that somehow memory formation is putting a heavy load on this brain structure. Now, a part of the brain getting bigger does not necessarily mean you're getting new neurons, but in fact, do we get new neurons in our brain as adult humans? Can someone answer?
No.
No. So the correct answer to first order is no. The more recent correction to that is, in fact, there are two small regions of the brain in which new neurons are born. One is near the olfactory area, leave that aside. The other one is in an area of the hippocampus known as the dentate. So we do get new neurons in the dentate area of the hippocampus, nowhere else in the brain. But this increase in volume is not necessarily due to the number of neurons. I'll come back and discuss what it could be due to. Now, there are correlate experiments to this that were developed about the same time, in the 90s, actually, when Morris developed his water maze, in which they put rats into kind of a kiddie pool. They filled it with water, and then they added, I did these experiments myself, they added powdered milk in there. So the water became kind of murky. And then they would put a platform in this water, a small platform somewhere in the middle. So you could locate the platform anywhere. The animal could not see the platform from the surface. You drop the animal in some corner of the pool, and the animal starts swimming until it finds the platform, right? And in the first trial, it swims a lot. And you're recording the animal swimming with a video that you've mounted in the ceiling, so you can kind of track the animal. In fact, my first lab, which was in Boyden Hall at Rutgers, I shared with Michael Ritchie, and one of the first things that we put there was one of these water mazes and a camera there. He was the one who was doing the research on the hippocampus. And I'll come back and talk to you about Michael Ritchie at a different session when I'm talking about the hippocampus. After the animal does this swimming for many trials, if you drop it into the pool, it swims directly to the platform and stands on it, because then it's safe, it doesn't have to swim and waste energy. Now, if you lesion the hippocampus bilaterally, so you basically destroy the hippocampus on the two sides, you will find that the animal will never learn. So even after days of trials, it does the same swimming to find the platform. Its learning is impaired for the same reason that it was in Henry Molaison, in that he could not form new memories, right? So whatever the animal learned, it was there for only a couple of minutes and then it's gone. So there's no long-term memory that is formed. All right. Interestingly, even if you hide all the cues that are around here, you see these visual cues for the animals, even if you hide them, people found that the animal can still find the location of the platform if the hippocampus is intact. Okay, is this experiment clear to everybody? If not, please ask. Okay, you got it, okay. All right, it's not a hard experiment, but what I want you to remember is that what we don't know from this experiment is whether the animal is unable to form memories or whether the animal is able to form memories but unable to store memories for the long term. Okay, there's somewhat of a difference there. Let's go back to the idea of what memories are. And I'm going to come back in, I think, a couple of lectures after Professor Friedman and discuss this in more detail. So in about late 40s, I think, Donald Hebb, who was a Canadian psychologist, published a book in which he proposed a neural mechanism for memory formation that was based entirely on the properties of synapses. And it's a very simple idea. He said, basically, neurons communicate through synapses. A synapse has a strength. It's how well a neuron can communicate its electrical activity to the postsynaptic neuron. All right? Now, if you have two neurons that make a synapse, he proposed that if these neurons are continuously active together, so neuron A and B are active simultaneously, more and more and more, then the synapse between the two gets strengthened. So if the activity of neuron A is happening at the same time that neuron B is excited and therefore active, that synapse gets strengthened. However, if these neurons are active at different times, they're not synchronous in their activity, they don't coincide in their activity, they do different things, then the synapse between these two neurons is weakened. This we now call the Hebbian learning rule for synaptic plasticity. Synaptic plasticity simply refers to the change in the strength of the synapse. How strong are the neurons communicating with each other? This was in 1949. In 1973, Terry Bliss was a graduate student who decided to go to the lab of Per Anderson in the University of Norway in Oslo. And Per Anderson worked on different areas of the brain, but in particular in the hippocampus. And Bliss paired up with another researcher in Per Anderson's lab named Thierry Lomo. And what they discovered, it's a really beautiful paper, this paper. It's very simple, and part of the beauty of this paper is how simple it is. They took slices of the rat hippocampus. These slices have a very, very distinct organization of cells. So if you slice the hippocampus, put it under the microscope and look at it, you see the regions of the cell bodies as you see here. The areas that you see in the hippocampus, if you look here, the main areas I want to point out to you here are area CA1 and area CA3. And what they looked at was the projections that come from the neurons whose cell bodies are in area. In fact, in this case, it's shown in the dentate. I think they looked at CA3 neurons projecting to the CA1. Okay, so dentate neurons project to CA3, CA3 neurons project to CA1, and I'll show you that in a few slides. But any of these synapses, if you look at it, what you can do is you can put, the stimulating electrode, you can literally put two wires there and activate the axons that are going to the postsynaptic cell. So in this case, they're showing axons being stimulated, coming from this pathway from the entorhinal cortex into the hippocampus and stimulating neurons that are sitting in the dentate. So you're recording a synapse from the perforant pathway axons to the neurons in the dentate gyrus. You stimulate, let's say, once per second. Every time you stimulate, you get an excitatory postsynaptic potential. You do this, in fact, it's less than once per second. I think it's less, one per like 2 or 3 seconds, okay? And what you do is you stimulate for about, let's say, half an hour, and you record the synapse. And in fact, the synapse is so strong if you stimulate because you're recording a population of synapses at the same time. These are excitatory synapses. Then each dot, imagine each dot corresponds to a synapse. Ignore the yellow one for now. You're looking at the green one, okay? So you're measuring the strength of the synapse, and whatever strength it is, you normalize to that. So you call that 100%. At some point, you produce a tetanus stimulus in this pathway. A tetanus stimulus is simply stimulating at a very high rate for a few seconds. In fact, you do a few tetanus stimuli. So let's say you go at 100 hertz for about a second, and then silent a second, another tetanus for a second. You do that a few times, let's say 6 times. And then you go back to stimulating once every 2 seconds and measure the strength of the synapse. The tetanus itself is not shown here. It's just shown by arrows. What you find is that this synapse is now almost 60% stronger. You put another tetanus, it gets stronger. Another one, it gets stronger. Another one, it gets stronger. This strength remains as long as you record it. So this is 6 hours that they're recording. Of course, at the time that these guys did it, recording extracellularly, you can keep this slice alive for 6 hours. If you record with a patch electrode intracellularly, you can't keep a cell alive that long. But as long as you could keep it alive, they saw this. This is the neural correlate of Hebbian plasticity. And I'll come back and explain it to you. They call this long-term potentiation. Potentiation is simply the strengthening of the synapse. Now, potentiation has a counterpart in the hippocampus, and that is called long-term depression, in which producing a different set of stimuli, for example, stimulating the presynaptic pathway with a lower frequency stimulus, not a tetanus, right? So you go 3 times per second, boom, 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 boom. You stimulate at that rate for about a minute. That leads to a weakening of the synapse. So the strength of the synapse goes to about 70% or 80% of what it was, right? Whereas LTP, it goes up by a certain percentage. Not all synapses get strengthened or weakened by the same amount. This is what is shown schematically in what happens in our knowledge today in long-term potentiation and depression. What you see here is in the postsynaptic membrane, you see the receptors for the neurotransmitter that is being released. The neurotransmitter that is being released in this case, does anybody know what the neurotransmitter that is exciting the neurons in the brain is? The primary one?
Acetylcholine?
Yes, no?
Is it
Primary excitatory neurotransmitter in the brain.
GABA? Glutamate?
No?
Acetylcholine?
Okay. Should I call on someone or no? Someone should hazard a guess.
Dopamine?
All right, Camila.
Can you hear me?
Yes, I can now.
Oh, I said acetylcholine.
No, sorry, but thank you for guessing. I'm not trying to put you on the spot, but acetylcholine is the primary excitatory neurotransmitter in the neuromuscular junction within the central nervous system. Michaela?
I was going to say glycine.
No, glycine is an inhibitory neurotransmitter. It doesn't excite. Colette?
GABA.
GABA is also an inhibitory neurotransmitter, but you guys are kind of going around, so there's only one big neurotransmitter left.
Glutamate.
Glutamate, yeah, thank you. So glutamate is the primary excitatory neurotransmitter. What you see here are receptors for glutamate. In fact, what you see are called AMPA receptors. During long-term potentiation, the synapse gets strengthened because AMPA receptors are added to the postsynaptic side, and the ones that are there get phosphorylated. What you see in this P is phosphate groups getting added to those receptors, which strengthens and stabilizes them. What you see in long-term depression is that the AMPA receptor gets dephosphorylated and it's taken out of the membrane. The process that it gets inserted into the membrane is in fact exocytosis. The receptors are sitting in vesicles right near the membrane and they just get pushed in. The process that it gets picked up is endocytosis. Okay, so by the way, the fact that not everybody immediately answered me on this question means that you should please review the synaptic section of cellular neuroscience, or I will record the lecture, please listen to it. I don't know if I'm not actually going to cover the different types of neurotransmitters and their receptors, but perhaps I should if I come back and I got a note that students are talking, but I can't hear them. Maybe I can't hear you because I'm talking over you. I apologize, but let me continue here. So long-term depression, long-term potentiation, the idea is pretty simple. You stimulate the synapse a lot, it gets potentiated. You stimulate it at a very low frequency, it gets depressed, okay? What is not clear yet is what did this have to do with Hebbian plasticity? Because you're only stimulating the presynaptic axons. You don't know much here about what the postsynaptic cell is doing. We do now. In fact, what we know is that long-term potentiation, and I'll come back and cover this in about a week or so, long-term potentiation requires a glutamatergic synapse to be active simultaneously with the postsynaptic cell being excited. In this case, let's call it being depolarized or producing an action potential. So for example, if you stimulate the presynaptic axon, so let's say you have two axons here, you stimulate one of them with a high rate stimulus, so that's a tetanus stimulus, that excites this guy, and the postsynaptic cell produces an action potential. So this A1 axon is simultaneously active with the postsynaptic cell because it's caused an action potential. Then this synapse becomes potentiated. The synapse that is nearby, because you didn't excite it, does not get potentiated, even though the postsynaptic cell fired. So if it is not active together with that, it is not potentiated. I apologize, I'm going to go back. This is what you see in yellow here. In yellow is a control pathway that you're just stimulating once every 2 seconds, but you're not producing a tetanus stimulus in that pathway. And as you can see, producing a tetanus in one pathway does not potentiate both of the pathways. It only potentiates the pathway that was active at the same time as the postsynaptic cell. That property is called selectivity. Only the pathway that is active together with the postsynaptic cell becomes active, okay? However, there is also what's called cooperativity and associativity, in that you can actually stimulate, so let me go here, you can stimulate one pathway and excite the postsynaptic cell. And if the second pathway is active at the same time, even if it's not a tetanus, that also gets potentiated, okay? So that's associativity. Cooperativity means that you need to have a cooperation between the presynaptic activity and the postsynaptic activity, or at least the voltage going up in the postsynaptic cell, okay? I'll come back and cover long-term potentiation, I told you, in about a week, but let me come to one other point which we now know, and that is a form of long-term potentiation in depression, which depends on exact timing of spikes between the pre and postsynaptic cell. And we call this spike timing dependent plasticity. So suppose you have a presynaptic cell and a postsynaptic cell and a synapse. If the presynaptic cell is active before the postsynaptic cell, always, so you activate the presynaptic and then post, activate the pre-post, pre-post. You keep repeating this, then the synapse can potentiate, its strength gets bigger. If you have the opposite, if the postsynaptic cell is active first, then the presynaptic, post first, then the presynaptic, they're going out of order for that synapse. It's the postsynaptic that's active first, then the presynaptic cell, then the synapse gets depressed. However, the potentiation and depression, the amount depends on the distance in time, interval in time between the spikes in the pre and postsynaptic cell. All right? If you go too far in time, then you get no effect on the synapse. And in fact, if they're always simultaneously active, you pretty much always get potentiation. All right. Let me skip this slide, and hopefully I'll come back to this in synaptic plasticity and go back to memory. When people looked more carefully at memory formation, they realized in rodents, in rats especially, that were primarily studied, and now in mice that have been studied, and then in other animals, for example, in bats, I'll come back to that, they found that there are a group of cells, in fact, pretty much all of the primary cells in the hippocampus, that produce activity when the animal goes through space. Let's say the animal's moving through a complicated maze. This cell becomes active at a specific location in the maze. Okay, they called that a place cell. So let's say this, imagine this is a room that the animal is moving about, and these lines that you see is the path that the animal has taken throughout the room in an hour. Okay, so it's gone all over the place and explored. This particular single cell that you're recording becomes active primarily when the animal is in this location. It's active other places too, but it is, we say, coding for this location in space. It doesn't matter if the animal came in this direction, or the animal went fast, slow. The cell is sensitive to the location in space. We call it, for that reason, a place cell. Of course, different cells nearby may be active in different places. So place cells actually code different locations in space, and potentially they're almost like the GPS for the animal. Now, if you look at the projections to the hippocampus, you see a set of cells that do something very similar. So the projections that come into the hippocampus come from a part of the cortex known as the entorhinal cortex. And the cells in the entorhinal cortex that are projecting, instead of being active in a single place, they're active on a grid. So the cell is active here, 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 here. And you see it's a hexagonal grid that it makes. And it's a really beautiful discovery. So the animal, unlike a Cartesian grid, which should be kind of a square, it makes this hexagonal grid. I think of it as built out of triangles here, this triangle, this triangle, this triangle, etc., in which it's keeping kind of a grid on the map that is going to the place cells. Together, it is thought that knowing the place, knowing a grid, and knowing the head direction of the animal, which occurs elsewhere in the same structure, is sufficient to code the entire movement in space for that animal. Okay? Now, in looking at memory formation from a computational viewpoint, people have thought about the process of having many cells presynaptically project to many cells postsynaptically, but you have a combination of connections. So let's say cell A that is presynaptic connects only to cells 2, 3, and 4. Cell B connects to 1, 2, and 4. Cell B connects to 1, 2, and 3. Cell C to 1, 3, and 4, etc. Then you have what's called the synaptic weight matrix. By changing the weights of these connections, what you do is you produce a particular engram, you produce a particular memory. It is possible to do this, and people, of course, this idea comes from computer science, right? It's not from biology, but people drew the parallel because they realized that the way the hippocampus is structured, more or less, is that it gets its inputs from the entorhinal cortex, as I told you, through these axons called the perforant paths. The perforant pathway comes and synapses onto these neurons known as granule cells in the area of the hippocampus known as the dentate gyrus. These are the cells that we can get born in adult mammals, including humans. The cells of the dentate, their axons are called mossy fibers. They project to the CA3 glutamatergic cells or pyramidal neurons in the hippocampus, and the CA3 cells project through their axons, which are known as the Schaefer collaterals, to the CA1 cells. The CA1 cells project out of the hippocampus back to the entorhinal cortex, but to a different layer of the entorhinal cortex. So again, this is a schematic drawn here, entorhinal cortex. You see the perforant fibers coming in, projecting to the dentate, mossy fibers to CA3, Schaefer collaterals to CA1, and then projection back to the entorhinal cortex. That structure produces for you the matrix of synaptic weights that I'm talking about. All right. To what extent this idea is valid in forming memories is a huge area of research. The number of labs in the world, neuroscience labs, that work on these particular pathways, both in vitro and in vivo, is literally in the hundreds, if not in the thousands. So it's a huge area of research, and a lot of what I'm telling you right now, despite the fact that it comes from computer science, has been verified. People actually believe this is more or less the way it works. Not every kink has been ironed out, but that's the way the hippocampus seems to function, by changing the weights in the brain. Now, interestingly, I have told you this. In HM, the problem that that patient had, Henry Molaison, when his hippocampus was removed, was not that he had problems with his long-term memories. He had all his long-term memories intact. He had problems forming new long-term memories. So knowing this, somebody tell me, is the hippocampus, you think, the site of storage of long-term memories, or is it the site of production of new memories?
The production of new memories?
Absolutely, because his long-term memories were intact. So all I'm telling you is not really telling you where the long-term memories are stored. It's just telling you how this structure can contribute to it. By the way, don't go around memorizing different regions of the hippocampus. I'm not going to test you on who projected to what and what projected to whom and come up with the names. That's not my idea of learning stuff. So the question of where the long-term memory is stored turns out to be a bit, if not a lot, more complicated. So let's take back this question to earlier researchers. This gentleman, physiologist and psychologist named Carl Lashley, around the turn of the 20th century, so in the early 1900s, he basically decided to do a set of experiments. So this is way before we had Hebb or the long-term plasticity and whatnot. He decided to do a set of experiments in rats to figure out the location of long-term memories, right? So he trained the rats to actually navigate different mazes, and the mazes differed in terms of complexity according to how many portions the maze had. So if the animal enters here, has to go through this leg, down this leg, up this leg, down this leg. This is a more complex maze than this one. The animal just has to learn to go down there and get to the food reward that's sitting at the other end. This is the simplest one. You just have to go down here and find the food. Now, the animal that's navigating these, right, it still searches the whole maze. When the animal enters, it doesn't immediately go to the next door and find it. It has to learn that, right? So what Lashley did is he trained the animals on all the mazes. So the animals became very good at it. They could find their way through the maze very rapidly, okay? And they made very few errors in this, okay? They didn't get lost, they didn't get stuck. Let's call that an error. Then what he did is he went into the brain with a scoop effectively and started scooping out different parts of the brain. And what he found, he started lesioning all different parts of the brain, different components of the brain, all right? So what do you think he found? Did he find that different regions of the brain are coding for different types of memories in this case? That seems like a reasonable assumption, right? So different, if the animal is learning one thing in particular, you expect it to be happening in one portion of the brain that eventually, if you take it out, maybe bilaterally at least, it would damage the animal's ability to do that action. In fact, what he found was a lot more surprising. What he found was that destroying, so in fact, what I should also say, he was going and destroying the cortex. What he found was that destroying different parts of the cortex does affect the animal's ability to go through the mazes, especially the more complex mazes, as is shown in this graph. Maze 3 is the one that produces the biggest effect in memory, and the higher this number, the more errors the animal is making. But it didn't really matter where in the cortex he was destroying. It mattered how much of the cortex he destroyed, right? So he came up with this hypothesis that memories are distributed all over the cortex. Okay, he didn't know how, but that was his hypothesis from this experiment. All right. So what do we know now? So in fact, we know that if you look at the projections from the hippocampus out to the cortex, they're projections from the hippocampus to a lot of the cortex, right? But especially in areas of the cortex known as association cortices or to the primary sensory cortices. Okay, we'll talk about the different parts of the cortex later, but there are projections from the hippocampus to all parts of the cortex. And interestingly, there are projections back from those parts of the cortex to the hippocampus. All right, so this indicates that there's some sort of communication between the hippocampus and the cortex that is essential in forming these long-term memories. These are declarative memories, memories in which you actually learn something and put it in place. You learn to do something and you have explicit access to it. We also know some of this, a lot of this in humans, for the different visual modalities, that memory of visual things gets stored mainly in the visual cortices, which are mainly in the occipital lobe in the back of the brain. Whereas, let's say, memories that are associated with sound, with auditory cues, get stored in the auditory cortices. These are the auditory cortex 1 and 2. We'll discuss this when we talk about the auditory system. And so different sensory modality memories can be stored in the corresponding cortical area for that sensory modality. So there is some division of labor, right? Except when you think about a memory, you rarely have a memory of anything natural that only happens through a single sensory modality. You see, you hear, you smell, you're in the environment, you get all your senses, often you touch the thing, and all of these are sending sensory informations that get integrated in the association cortices, okay? And therefore they get stored in the same cortices, which are kind of distributed in different parts of the brain. I'm going to come back and talk about this in a little bit, but non-declarative memories, which are the implicit memories, apparently involve different regions of the brain that are not the cortex. These are the deeper parts of the brain, well, they're parts of the cortex, but the part of the cortex they include are a part known as the prefrontal cortex. I'll discuss that in a bit. But they also involve the amygdala, some of the sensory association cortices, the basal ganglia especially. So the basal ganglia, especially a big part of the basal ganglia, the main nucleus, which is known as the striatum, and the cerebellum, which is this part in the back of the brain. So those are for implicit memories, such as motor skill learning and manual tracking of things, tracking a spot of light, tracing a curve, using a mirror, reproducing a sequence. These are all implicit memories. Now, in people who have damage to their basal ganglia or get a disorder that deteriorates, let's say, the striatum, for example, Huntington's disease does that. This is the type of memory that gets destroyed often. All right. So this I already told you, that the visual cortex becomes activated in vivid remembering of particular visual images. And it is interesting that different types of images produce activation in specific areas of the cortex. But not only does the image produce that activity, but if you actually think about that image, you get activity in the corresponding areas that you got when you saw the image. This is done with functional magnetic resonance imaging, which you can use to actually look at real-time activity in a living person, right, or a living animal, and look at where in the brain are neurons active. Okay, so declarative memory requires extensive input from the hippocampus. There's also regions of the brain that are specialized in specific types of declarative memories. For example, in language. So we know that this area known as Wernicke's area, if there's damage to that, people are unable to understand language. They don't understand words, but they can speak. They can use syntax, they can use grammar, they can intonate, they can produce the rate, they just don't understand the language. In contrast, if Broca's area, which is this region marked here, is damaged, people get the opposite problem. They understand language, but they can't speak and they can't write. They can often say individual words, but they can't actually produce it in a syntax, much less a grammar, and they can't produce the right intonations. Okay. What I want to say about language, so somebody tell me, why is language important in the topic of studying the brain. What is special about language?
Because it uses so many different parts of the brain, like you have to be able to understand it and translate it.
Anything that you can think of? Let me ask this a different way. Is language unique to humans?
Not exactly.
Let's go for it.
Sorry, I don't know if you could hear me before. But I was saying like it's not really unique to human, and the reason that it's interesting to study is because it uses so many different parts of the brain, like understanding it and putting meaning to it uses different parts of your brain. It's not just one aspect.
Good. So language, so what do we use language for, actually? Since you're there, tell me.
Communication.
To communicate. So, but if you think about it, language is somewhat more than just communication. Obviously, a lot of animals communicate. We know that, you know, from bird songs to fish to whales to, you know, definitely apes like humans. They communicate vocally, they communicate with signs that have to do with moving the body. A lot of animals, even insects, communicate with each other. Honeybees have dances that they communicate the location of food, flowers, etc. But language is a bit more than a tool for communication. All right, so maybe at some point we can come back and discuss language. I don't have a special lecture on language, but those of you who are honor students, it's an interesting topic to go into, because what we think as neuroscientists, and I would say that most biologists agree with this, despite the fact that nobody doubts that we are evolutionary descendants of the same animals as all the animals in the world, that language in a way is what we think makes humans as a species unique. So in this sense that I'm talking about language, where I talk about grammar and meaning, that, as far as we know, is unique to humans. And we can discuss this later. Okay, I just wanted to make that point here, but let me continue. All right, so let's go a little further back here. There's the process of learning. I also want to say that the process of learning can also be interfered with. You can interfere with memories, and interfering can result in forgetting, and if it's long-term effect on the on memory, it's amnesia. So interference happens when there's typically interaction between new material and past learned memories or behaviors. There's such a thing as retroactive interference, where new memories simply replace old memories. So if you ask me, what was your address 10 years ago, I may not remember it because, you know, why do I need to? So it just gets, you know, pushed aside and put away. And there's proactive interference, in which old memories block your ability to learn new information, as when you use, you know, your old girlfriend or boyfriend's name to call your new girlfriend or boyfriend. And even more embarrassing, you use your old wife's name to call your new wife, and then you get a slap or a dope slap. The correlates of this in amnesia, and we talked about this, are anterograde versus retrograde amnesia. And we know that anterograde amnesia is caused by faults in the hippocampal structure, whereas retrograde amnesia is a more complex thing that requires damage to cortical structures. So for example, Wernicke's area or Broca's area, those are cortical areas, and damage to those structures affects your ability to use language that you've learned in childhood effectively. All right. Now, this anterograde amnesia, yes, we have patients like HM and Cochrane, Brent Cochrane, or Eric, I don't remember his first name. Now I'm having retrograde amnesia there, but Can anybody give me an example of a problem in which retrograde amnesia occurs, or a disease in which, or disorder in which retrograde amnesia occurs pretty commonly now?
Dementia.
Dementia. And what
And
is the
Alzheimer's?
worst? Alzheimer's. So Alzheimer's is an aggressive form of dementia. And that's exactly the problem. So Alzheimer's is caused by, is strongly believed to be caused by these neurofibrillary tangles and plaques in the cortex. And it is the #1 neurological disorder in the developed world, and I think all over the world. And it's devastating because people lose their memory. Okay, so that's the hallmark of the pathology of Alzheimer's, that people start to forget things, and it gets progressively worse and worse and worse. And these are due to damage in the brain, and you can kind of see in this area, these are the pathological changes in the brain of an Alzheimer's patient. This is on the surface of the cortex in the temporal and occipital lobes, and this is in a sagittal cut. You can see in the other areas of the cortex where the amyloid plaques and the tangles have occurred. So we know the correlates that affect long-term retrograde, that produce long-term retrograde amnesia, can happen by damage to the cortex, as in Parkinson's patients. All right, I'm going to now take a pause. So far in the lecture today, I focused on the hippocampus, cortex, and declarative memory. Let me now take a step to the other side and talk about implicit memories and implicit learning. Okay, so these are the types of memories that typically don't depend on the medial temporal lobe and the hippocampus. Okay, implicit memory, you can divide it into two main categories. One is called non-associative learning or perceptual learning. The other one is called associative learning or conditioned learning. All right, so non-associative learning is really the simplest form of learning or memory formation, and it happens in every animal. No matter how simple the animal is, it has this particular response. Even I think in jellyfish, which, you know, a lot of people say jellyfish don't have a brain. They have neurons, they have like a neural ring, but they do show habituation and sensitization, right? And that is either responding less and less to a stimulus or responding more and more to a stimulus. And the stimulus is a sensory stimulus, okay? Associative learning is typically divided into two classes: classical conditioning and operant conditioning. And I'll come back and discuss those in turn. So habituation, here's an example of it. Here's a rat in which the animal is given an odor, and when it gets an odor, it goes and investigates the source of the odor, and it spends some time there. You give it another odor, it investigates again, but for less time. Again, you repeat it, it investigates less. So the odor is coming with the same plume at the same amount, but the animal investigates less and less and less. It is habituating to the odor response. You give it a different odor, it goes back and investigates again with the same vigor as the first time, okay? So habituation is a behavioral learning paradigm, is a behavioral type of learning, but it is odor specific. Habituation is stimulus specific. It's important to remember this. Conditioned learning, I told you, is divided into classical conditioning, which is also known as Pavlovian conditioning, which basically is associating an involuntary response to a stimulus that happened before an experience, okay? And operant conditioning, which is changing the behavior as a result of changes in memory and changing in your behavior as a result of experiences that happen after the response. Let me tell this in a bit more detail. Classical conditioning, we pretty much have all seen in Pavlov's dog. You give an animal an unconditioned stimulus, you get an unconditioned response. So you show the animal food, you see it drooling, you see it salivating. Now, what you do is at the same time as you show the unconditioned stimulus, you show a conditioning stimulus that is neutral. For example, you blow a whistle or you ring a bell or you produce a light, right? And you repeat this several times. And what you get after the conditioning happens is that simply producing the neutral cue, which is the conditioning stimulus, produces the response. So after conditioning, simply the whistle can cause salivation. Again, so long as the animal can detect the stimulus, any stimulus can be a conditioning stimulus. Now, I cannot emphasize this enough. Conditioning happens all the time to us, to all animals, as we go around our business every day in the world. Conditioning is our primary default way of learning. That's how you form memories. You associate things. That's what the brain's job is mainly in learning. Most of the learning in the brain happens through implicit learning, which is habituation, sensitization, and conditioning. All right. Now, in this case, the animal didn't have to do anything. It just got cues, and the cue became a memory, right? That's classical conditioning. In contrast, if the animal has to do something to actually learn, that becomes operant conditioning. But before I go there, let me come to an example of this in a simpler animal. And I'm going to come back to this in about a week when I talk about synapses again. This animal that you see pictured here is a sea slug. Sea slug is just like a land slug, but this particular sea slug, there's a lot of different types of sea slugs. They're kind of slugs, right? It's just the snail without its shell. Think of it that way. A soft-bodied invertebrate animal. This particular one is called Aplysia californica. It's indigenous to the Pacific waters of California, and they're very big. They're really, really big animals. So the Aplysia breathes through a siphon that it pushes out of its back. And when the siphon is pushed out, it's kind of like a tube, right? It pulls out the gill of the animal too, right? So the gill and siphon are out when the animal is going about its business under the ocean. If you touch the siphon, the animal pulls it back. Okay, so a tactile stimulus allows the siphon to be pulled back. If you keep touching, so you touch the siphon, the animal pulls it in. If you don't touch, it pushes it back out after a while. You touch it again, it pulls it in. It pushes it back out. You touch it again, it pulls it in. After a while, it realizes that this is an innocuous stimulus. So after 13 times of you touching it, it stops pulling in the gill because it's not producing any bad effect on the animal. Now, what you can do and what people have done on this animal, and it resulted, in fact, in a Nobel Prize to Eric Kandel, because this was the place that for the first time an engram was shown incontrovertibly in this animal. If you touch after the animal has habituated, you touch the siphon and at the same time you shock the tail of the animal with an electrical shock, or you just pinch the tail, right, to produce pain. You do that once, the animal immediately withdraws the gill, right? And if you do that 4 times, let's say you do it once with the shock, you wait about 5 minutes, you do it again, 5 minutes, 10 minutes, you do it again, you do it 4 times, then you go back to the touch alone, right? So even though the touch alone is innocuous, the animal every time will pull its gill in. And it will do it for that entire rest of the day. So that memory lasts for a day. You do that over 4 days. You pair the shock and the touch 4 times over 4 consecutive days, okay? That's in the, I think I'm going to show that. This behavior, this learning paradigm is called the gill withdrawal reflex, or sometimes it's called the siphon withdrawal reflex in aplysia. All right. Now, all that is involved here, and I'm going to come back and walk you through it in a week, is that the gill is simply innervated. I'm sorry, the siphon is innervated with a sensory neuron that makes a direct synapse to a motor neuron that pulls the gill back in. So it's a sensory motor reflex. All right. The touch activates the motor neuron, pulls the gill in. Shocking of the tail activates a different sensory neuron, a noxious receptor, a nociceptor, that then activates a neuron that releases serotonin. 5-HT is serotonin. Serotonin goes in and modulates the activity of the sensory neuron, and especially it modulates the synaptic strength between the sensory neuron and the motor neuron, and makes it a lot stronger. It produces long-term potentiation of that synapse. And that is how the innocuous sensory input, even without the tail shock, can produce the withdrawal of the gill, because the synapse now becomes so strong. This is what I was telling you before. If you give a control stimulus, then the animal responds then, but then it doesn't respond the days after. These are 7 days after training. If you give the animal 4 single shocks, it will remember it for about a day and then it'll forget. If you give it of 4 trains of 4 shocks, then it will remember it even longer, even after 4 days or 7 days, it remembers it. And if you do this 4 days in a row, then the animal will never forget. It just remains there. And what you know is that this change in behavior or the lasting of the memory is correlated with the strength of the synapse being modulated by serotonin. Okay, we'll come back and discuss this in a bit more detail. Right now, I just wanted you to see that formation of long-term implicit memories happens in all animals, and we have an idea of how it could happen. In this case, through neuromodulation by serotonin. The cellular mechanisms underlying that, I'm going to come back and discuss, so let me skip that. Let's go back to conditioning, the other side. Operant conditioning is a response to a behavior associated to a voluntary behavior. So for example, the animal gets a light. If it gets a green light, then it has to move a lever and it gets food. Okay, if it gets a red light, if it touches the lever, it's shocked. The animal learns that it should only move the lever when it gets a green light, and it learns it very fast if you give it a shock with a red light. If you don't give the shock, it takes it a lot longer to learn. Now, these experiments, rats are great at these. They learn really fast. This is an operant conditioning paradigm. You have to do something to learn. It's active conditioning, okay? Conditioning, both in the Pavlovian sense, classical conditioning, and operant conditioning, are the main type of learning that happen, for example, during addiction. If you take my addiction class, or if any of you took my addiction class in the fall, I talk about the essential centrality of this type of memory formation in addiction. That's all it is, in fact. It is conditioning. And if you haven't taken my class and are interested in the topic, take it next fall, and hopefully, and I will tell you all about it. All right. a lot of what we learned about operant conditioning came through the work of this gentleman. He was another neuropsychologist, B.F. Skinner at Harvard. And in the mid-90s, he did a set of experiments both on rats, on rodents, and on pigeons. And pigeons turned out to be quite smart in that, for example, in this particular case, the pigeon is in a box. It has 4 dots in front of it with 4 different colors. It gets a picture that has a specific color. It has to pick that color and peck on that. And then in response to that, it gets a few grains of seed as food reward. And the animal learns to do that very well. Now, the modern versions of this happen with, let's say, a rat in a box. I showed you this in the previous example. There's a light that comes on. There's an auditory cue that could happen. The animal has to move the lever to get a reward. And if it does things wrong, then it gets a foot shock through this electrified grid. This is known as a conditioning chamber, or more endearingly as a Skinner box, named after B.F. Skinner. Watch this video on operant conditioning and free will. It's linked in the PDF that I put for this lecture in the Canvas page. And watch also what he says about what he thinks about free will from doing these conditioning experiments. Let's do a quick vote here since I'm on the topic of free will. This is not a discussion. This is just, raise your hand if you believe in free will. Do we have a choice in what we do? Do I have a choice in things that I do in my life? Or is it all predetermined? If you think I have a choice, raise your hand.
I could curse you out right now, but I'm not because I have free will.
Ah, you got me, man. That was a good one. All right, so, free will is getting a good majority. So we have a good majority that believe in free will and have a healthy minority that are pessimistic brats like me who don't believe in free will. Okay, and we can stick to our belief, but I want you to know what B.F. Skinner says about it. Okay. All right, onward. What we know about this kind of learning in mammals is that implicit learning and all types of conditioning require dopamine. Okay, in what sense? In what sense do they require dopamine? I took this picture, by the way, all the pictures that I take that I don't give citation to are from Wikicommons or Wikipedia. Those are on public domain. Okay, so every picture that you see here that I haven't written, this one I wrote to remind me to tell you this. Everything that you see that I don't, put a citation on is either from Wikipedia or from our own textbook. Okay, I don't have to cite my own textbook because I'm using it for class. I love Wikipedia. I think Wikipedia is one of the greatest advances in human knowledge. I pay money to them every year, and I think it's a great, contribution to humankind. However, if you ever cite Wikipedia for a piece of knowledge, I will give you a 0 on that assignment. Wikipedia is not for citation. It's for you to go as a first step and learn about things. Okay? Reinforcement learning is a phrase that comes again from computer science. Now, in modern lingo, we call this area deep learning or artificial intelligence sometimes. Basically, you can think of a piece of computer code or a part of your brain that acts as an agent that is allowed to take an action and produce an output. So taking an action means doing something in the environment, but let's say you produce an output and you see the result of that output, you observe that. Let's say, in your case, some part of the brain makes you do something and you observe what you did, you see the result of what you did, and that observation allows you to kind of represent the state of that action back to the agent by giving it a reward or not giving it a reward or reducing the amount of the reward. If the agent is doing something that you, by observation, see is useful, that is going in the correct direction, you give it a reward. If it's going in the incorrect direction, you don't give it a reward. By doing this, you reinforce the ability of the agent to do that action, and this becomes a feedback process that produces learning, right? There are algorithms in computer programming that you can write that do this, do reinforcement learning. And in fact, it is a big part of what is known as unsupervised learning. You simply have an agent, have an algorithm that does things by trial and error, except you reward the ones, you make the ones that are correct happen more and more and more. And as they become better and better, you keep picking those. So I don't have to tell it what is correct or not. I just have to keep giving it a reward. Okay? So in our brain, we believe we have a similar mechanism. And in fact, we being people who study this area, I'm a novice to this area. I started doing some research in this area of dopamine just about the year before COVID, or half a year before COVID, and I'm continuing this, but I'm not an expert in reinforcement learning. But I will tell you what we know and what we think is happening. What we know is that there's an area in the midbrain. So this is the brainstem. There are a couple of nuclei in the brainstem known as the dopaminergic nuclei. And in particular, there's one known as the ventral tegmental area and one known as the substantia nigra. These areas project their axons to primarily parts of the striatum, ventral or dorsal striatum, but they also project to the cortex. In VTA projects to the cortex, especially prefrontal cortex, and to parts of the hypothalamus. We think that learning that is involved in conditioning processes always require dopamine release from these two areas. If you lesion the dopaminergic neurons in the animal, it will not be able to learn the new tasks anymore. So reinforcement learning requires dopamine. And in fact, in Parkinson's disease, what happens is that you lose the dopaminergic neurons specifically in the substantia nigra. And in the patients who have Parkinson's disease, you can do this test in that, so think about having 3 doors that are colored, and you learn that, let's say, knocking on door pink gets you a reward, and this is a computer test. But you put another stage that you have to knock on door red to get to this stage. Knocking on other doors doesn't get you there. And here, knocking on door white to get to this stage. Knocking on door yellow gets to this stage. If you're a Parkinsonian patient, you can learn the early stages well, but as you go back and back, you can't learn that. You can't learn the 4 stages. Whereas the control people can learn this perfectly, going through this. You and I can do this perfectly. Take the Parkinsonian patient, put him on L-Dopa, and they do almost as good as the control case. Recovering the ability to learn requires simply dopamine production. L-Dopa stimulates producing dopamine. All right, so what did I tell you? Declarative and non-declarative memory are stored differently in the brain. Declarative memory requires the hippocampus for formation and producing short-term memories, but is stored primarily in cortical areas. Non-declarative memory has many sites potentially of forming, including the striatum, basal ganglia, and the cerebellum, and perhaps part of the limbic cortex, and especially another region that I keep forgetting to mention is the amygdala, and the sites of the long-term storage are clearly the basal ganglia and the cerebellum and parts of the cortex, including prefrontal cortex and the premotor cortex, which we will discuss later on. Okay, we are at the end of class time. So I'm going to skip this last slide and I will see you guys. I actually want to see you guys. Wait, next Monday is in person, right? So Professor Friedman will do the lecture on Monday. Thank you all.
Have a good day.
Have a good day.